So now that we have our purified egfp inserts, the next step is going to be to try to insert this egfp gene into the PET41A plasmid. We see, however, that since we digested our PET41A plasmid with the restriction enzymes, the NCOI and the NOTI restriction enzymes, we have restriction sites here, so ends that have been cut here, creating these sticky ends. However, the egfp gene has blunt ends, so we actually can't ligate it together. And so what we're going to have to do is we're going to have to digest our egfp inserts with the same restriction enzymes so that they can get sticky ends. And so then now we can insert the egfp insert into our PET41A plasmid, and so it can actually fit almost like a puzzle piece. So in order to do the restriction digest, we're going to want to start off with the Eppendorf tube, and we're going to be adding in our egfp inserts. And so if we were to look at one of these inserts, it'll look like this. And so we're also going to be adding in CutSmart buffer and sterile water. But the important components we're going to be adding in is our restriction enzymes. And so we see that whenever we did the egfp amplification process, the primers we used were actually really important because these primers were now embedded within the egfp insert. And because of that, these primers have been created to have sites that are engineered to be restriction enzyme sites. So essentially, these primer sequences were embedded into our egfp insert. And these sites are actually able to be recognized by our restriction enzymes. So for example, we see that whenever we have our NCOI restriction enzyme and our NOTI restriction enzyme, they're going to recognize these sites here within our plasmid, within our egfp insert. So for example, the NOTI restriction enzyme will recognize this segment here and will cut here. And the NCOI restriction enzyme is going to be able to recognize this site here, and it's going to be able to cut here. And so because of that, once we digest this egfp with the restriction enzymes, our end product is going to be the digested egfp insert. And so now we're going to see that once we have that, we will then be able to insert that egfp into our PET41A plasmid.